Welcome friends to a new session on molecular biology. In this session, we will discuss processing of ribosomal RNA, rRNA, and transfer RNA, tRNA. Processing of ribosomal RNA and transfer RNA. The tRNAs and rRNAs are the structural molecules that have very important roles in protein synthesis. Ribosomal RNA, rRNA. In eukaryotes, the 4 rRNAs are first transcribed as 2 long precursor molecules. One of the precursor molecules contains the pre-rRNA that will be processed into the 5S rRNA, and the other contains the 5.8S, 18S and 28S rRNAs. The enzymes then cleave the precursor molecule into subunits corresponding to each rRNA. In prokaryotes, there are only 3 rRNAs and all are transcribed in one long precursor molecule that is cleaved into the individual rRNAs. Mature rRNAs make up 50 to 60% of each ribosome. Some of the ribosomes RNA molecules are purely structural, whereas others have binding or catalytic activities. The eukaryotic ribosome is composed of 2 subunits, a large subunit 60S and a small subunit 40S. The 60S subunit is composed of the 28S rRNA consisting of 3354 nucleotides, 5.8S rRNA consisting of 120 nucleotides, 5S rRNA consisting of 154 nucleotides and 50 proteins. The 40S subunit is composed of the 18S rRNA consisting of 1753 nucleotides and 33 proteins. The prokaryotic ribosome is composed of 2 similar subunits with slightly different components. The large subunit is the 50S subunit, which comprises of 23S rRNA consisting of 2839 nucleotides, 5S rRNA consisting of 122 nucleotides and 31 proteins, and the small subunit that includes the 30S subunit is composed of the 16S rRNA consisting of 1504 nucleotides and 21 proteins. The 2 subunits join to constitute a functioning ribosome that is capable of creating proteins. Transfer RNA, tRNA. Transfer RNA having a mass of about 25 kilodaltons consists of about 75 nucleotides, which makes it one of the smallest of the RNA molecules. Transfer RNA carries amino acids in an active form to the ribosome for polypeptide bond formation in a sequence dictated by the mRNA template. Each different tRNA binds to a specific amino acid and transfers it to the ribosome. There is at least one kind of tRNA for the 20 amino acids. Mature tRNAs take on a 3-dimensional structure through intramolecular base pairing to position the amino acid binding site at one end and the anticodon in an unbase paired loop of nucleotides at the other end. The anticodon is a 3-nucleotide sequence unique to each different tRNA that interacts with a mRNA codon through complementary base pairing. There are different tRNAs for the 21 different amino acids. Most amino acids can be carried by more than one tRNA. Transfer RNA is the adapter molecule in protein synthesis. mRNA acts as the template for protein synthesis. How then does tRNA direct amino acids to become joined in the correct sequence to form a protein? In 1965, Francis Crick wrote, RNA presents mainly a sequence of sites where hydrogen bonding could occur. One would expect, therefore, that whatever went onto the template in a specific way did so by forming hydrogen bonds. It is therefore that a natural hypothesis that the amino acid is carried to the template by an adapter molecule and that the adapter is the part of that actually fits onto the RNA. In its simplest form, one would require 20 adapters, one for each amino acid. This highly innovative hypothesis soon became established as fact. The adapter molecule in protein synthesis is tRNA. The tRNA contains an amino acid attachment site and a template recognition site. A tRNA molecule carries a specific amino acid in an inactivated form to the site of protein synthesis. The carboxyl group of these amino acid is esterified to the 3' or 2' hydroxyl group of the ribosome unit at the 3' end of the tRNA chain. The amino acid joined to a tRNA molecule to form an aminoacyl tRNA is catalyzed by the enzyme aminoacyl tRNA synthetase. This esterification reaction is driven by ATP cleavage. There is at least one specific aminoacyl tRNA synthetase enzyme for each of the 20 amino acids. The template recognition site on tRNA contains anticodon, and this anticodon recognizes a complementary sequence of 3 bases called a codon on mRNA. Structure of tRNA proposed by Alexander Rich and Aaron Klug. The tertiary structure of tRNA was first reported in yeast tRNA phenylalanine, was determined independently by Alexander Rich and Aaron Klug in 1974 using X-ray analysis of tRNA phenylalanine crystals. They demonstrated that the main pattern of tRNA chain folding to tertiary structure is universal. The folding takes place by the following manner. The acceptor stem and the T arm are arranged along a common axis, forming a continuous double helix which is 12 nucleotide pairs length. The anticodon arm and dihydrouridylic arm are also arranged along a common axis and yield another double helix which is 9 nucleotide pairs long. These two helices are oriented towards each other at approximately at right angle, so that the dihydrouridylic loop is brought to the closed T loop. As a result, the structure looks like the letter L. At the top, its two limbs corresponding to the anticodon and the acceptor 3' end. The short single stranded bridge between the acceptor stem and dihydrouridylic helix, part of the dihydrouridylic loop, and the additional variable loop are superimposed on the dihydrouridylic helix in the region of the inner corner of the L-shaped molecule, resulting in the formation of the core of the molecule with a number of tertiary interactions.